What if you could change the instructions for life itself? What if you could tell a plant to grow bigger, or tell a living cell to fight off a disease? It sounds like science fiction, but it's a real field of science happening right now. It's called genetic engineering. Imagine a farmer whose entire field of corn is being destroyed by a pest. The farmer needs a way to protect the crops without using harmful chemicals. Or think about a person born with a disease caused by a tiny error in their body's instruction manual. These are huge problems that are incredibly difficult to solve from the outside. For centuries, we could only observe these problems. We could treat the symptoms, but we couldn't fix the root cause. The cause is hidden deep inside, written in a code that every living thing shares. That code is DNA. To understand genetic engineering, you first have to understand DNA. Think of it as a giant cookbook found inside every single cell of an organism. This cookbook doesn't have recipes for cakes or cookies. It has all the recipes for building and running an entire living thing. The recipes are called genes. One gene might have the instructions for your eye color. Another has the instructions for how your body digests food. These instructions are written in a simple four-letter alphabet, A, T, C, and G. The specific order of these letters is what makes a rose different from a beetle, and a beetle different from you. Genetic disease is like a typo in the cookbook. A single wrong letter in a critical recipe can cause a big problem. A weak plant might be missing the recipe for defending against pests. The instruction just isn't there. So scientists asked a powerful question. What if we could become editors of this cookbook? What if we could fix the typos? This is the core idea of genetic engineering, changing the DNA of an organism on purpose to change its traits. For a long time, this was incredibly hard. The DNA cookbook is billions of letters long, and finding one specific sentence to change was like finding a needle in a giant haystack. Scientists first developed methods to cut a gene from one organism and paste it into another. For example, they found a gene in a bacterium that produces a natural insect repellent. They figured out how to carefully snip out that gene and insert it into the DNA of a corn plant. Now, the corn plant had a new recipe. It could create its own protection against the pests that used to destroy it. This is called creating a genetically modified organism, or GMO. But this cut and paste method was often clumsy and not very precise. Then, a revolutionary new tool was discovered, inspired by the immune system of bacteria. It's called CRISPR. Think of CRISPR as a pair of molecular scissors combined with a GPS. Scientists can program CRISPR to find a very specific sequence of DNA letters in the vast cookbook. Once it finds the exact spot, it can snip the DNA with incredible precision. This allows scientists to not just add new genes, but to delete unwanted ones, or even rewrite a specific letter, like correcting a typo. It's like using a find and replace function on a computer, but for the code of life itself. This level of control opens up a world of possibilities that were once unimaginable. The big insight here is that biology isn't just something to be observed. It's a system that can be engineered. We are learning that DNA is not some mysterious, unchangeable force. It's a form of information. It's a programming language that nature has been using for billions of years. And for the first time in history, we are learning how to read, write, and edit that language directly. With this power, we can design solutions to some of our biggest challenges. We can engineer crops that grow in salty water or survive droughts. We are exploring ways to correct genetic disorders in humans, potentially curing diseases that were once a life sentence. Genetic engineering gives us a new way to solve problems, not from the outside in, but from the inside out, by rewriting the very blueprint of life.